In this module, we will discuss cluster analysis for high-throughput genomic data. As a reminder, we have already discussed the preprocessing methods including image analysis for array-based data and alignment for RNA-seq-based data, normalization, and expression summaries. Once we have expression summaries, we then perform our planned statistical analysis. We will now focus on the task of cluster analysis. There are two large taxonomies describing analyses of high-throughput genomic data that have been derived from the machine learning literature: supervised learning and unsupervised learning. Supervised learning incorporates knowledge of phenotypic information of the samples being examined. Modules 7 and 8 pertain to differential expression, which fall under supervised learning, because the class or phenotypic labels are being used in the analysis. Unsupervised learning refers to an analysis where phenotypic data are either not available or are not used in the analysis, and the goal is to use only the expression values to discover structure in the data, such as clusters. Since the goal of high-throughput genomic data analysis is to find relationships and patterns in the data and ultimately achieve new insights into the underlying biology, unsupervised learning methods have been readily applied. In general, searching data for a structure of natural groupings is an important exploratory technique. For high-throughput genomic data, we may want to cluster samples, or in other words, identify disease subgroups that may affect clinical outcome. Or perhaps we want to cluster genes, to identify genes that share similar biological functions, or identify genes co-regulated by the same mechanism. This figure produced using these data is a plot of height in centimeters by weight in kilograms for 8 individuals. Most would agree that there are two clearly identifiable clusters in the set of data. In fact, upon inspection of the additional data available for these subjects reveals that the two clusters are aligned with another explanatory variable, adult versus children. It is important to note, however, that if other associated measurements for these same study subjects were used in clustering them into homogeneous groups, such as birth order, birth month, IQ, whether they are left-handed or right-handed, we may have arrived at a totally different clustering of the same 8 individuals. Therefore, it is important to note that any clustering produced should be based on variables in which the investigator is interested, and that there are likely several different clusterings of the same set of objects. In 1993, Calgill listed the following general steps that are involved in a cluster analysis. The objects to be clustered must be selected. We can cluster observations or genes. The variables to be used in the cluster analysis must be selected. The variables must contain enough information to permit the accurate clustering of the objects. The proportion of relevant information to random error or noise should be kept to a minimum. For that reason, the gene expression dataset is often filtered. The researcher must also decide whether or not to standardize the raw data. Often, gene expression is centered by subtracting the mean or median. Additionally, a decision must be made whether to categorize continuous data into finite group. A distance or similarity measure must also be chosen. A clustering technique must be chosen. The number of clusters must be ascertained. The researcher must interpret the meaning of the clusters in terms of the research objectives. The difficulty in validating a cluster analysis derived classification is that there is usually no gold standard classification against which the clustering results can be compared. The data from a gene expression microarray experiment is typically stored in a matrix where rows represent genes or probe sets and columns represent samples. For example, in this data matrix, X sub 1, 2 represents the expression value of the first gene for the second sample. Gene expression data must be normalized and is usually log transformed. For RNA-seq data, this can be VUM normalized if using the Lima package, or RLog or VST transformed if using the DE-seq2 package. Typically, the resulting normalized gene expression values are not used directly in a clustering algorithm. Rather, these expression values are converted to a distance or dissimilarity or similarity measures, and the clustering algorithm is applied to them. It is important to determine what distance measure is the most appropriate for the data at hand before choosing a clustering algorithm. This is due, in part, because some clustering algorithms will only work for certain distance measures. Additionally, one must be willing to assume that all relevant relationships within a set of objects to be clustered can be summarized by a matrix consisting of distance dissimilarities between each pair of objects. Distances, dissimilarities, and similarities will all be referred to as proximities, which are the usual input into a clustering algorithm. We will first discuss distances and mention the criterion for a distance to be metric is that these following four assumptions must be met. Briefly, these properties are that distances are non-negative, the distance between a point and itself is 0, distances are symmetric, and the shortest distance between 2 points is a straight line. Euclidean distance is commonly used in many clustering algorithms. By definition, the Euclidean distance is defined by this equation. Geometrically, for two-dimensional data, the Euclidean distance is based on the Pythagorean theorem, a squared plus b squared equals c squared. The standardized Euclidean distance may be computed using either the variance or range. The Manhattan distance is also known as city block distance, since it corresponds to the distance one would travel to get from x to y over right angles. Specifically, Manhattan distance is defined by this equation. Both the Euclidean and Manhattan distances are specific cases of the more general Minkowski distance, also referred to as LP metric, which is defined by this equation. The parameter p is the order. When deciding on an appropriate distance, that is, the selection of p, the investigator must decide whether more emphasis should be placed on larger differences. Other distances include Mahalanobis distance, which is given by this equation, and Canberra distance. The dist function computes and returns the distance matrix computed using the specific distance measure to compute distances between rows of a data matrix. Methods available include Euclidean distance. Here you can see the distance between all pairs of observations. For example, the Euclidean distance between observations 1 and 2 is 69.836. The Euclidean distance between observations 1 and 3 is 2, while the distance between observations 4 and 6 is 26.401. Changing the method parameter in the dist function to Manhattan, the Manhattan distance between observations 1 and 2 is 95, the Manhattan distance between observations 1 and 3 is 2, the Manhattan distance between observations 4 and 6 is 37. When the method parameter in the dist function is Minkowski, you additionally need to specify the parameter p. When p equals 3, the Minkowski distance between observations 1 and 2 is 64.335. When p equals 0.5, the Minkowski distance between observations 1 and 2 is 186.082. The method parameter can also be set to calculate Canberra distance, which is sensitive to small changes close to 0, and a few other methods are also available. Gene expression profiles represent expression measures, and therefore Euclidean distance may not be considered to be that meaningful. Therefore, we need a distance measure that scores based on similarity between genes. This similarity measure should require that the more objects I and j are alike, or close, the larger S of I and j becomes, which leads to the following properties of similarity measures. The similarity is in the 0-1 interval, the similarity between an object I and itself is 1, and similarities are symmetric. Pearson's correlation, defined by this equation, is often used for gene expression data, though it must first be transformed to meet properties required by similarity measures. That is, you cannot directly pass Pearson's correlation estimates to a clustering algorithm. Remember, Pearson's correlation is on the -1 to 1 interval. Typical transformations are the quantity 1 plus correlation divided by 2 treats highly positively correlated genes as being similar, but highly negatively correlated genes as being dissimilar. And the absolute value of the correlation, which treats both highly positively correlated and highly negatively correlated genes as being similar. Selection between these two transformations must be based on subject matter knowledge, that is, whether highly negatively correlated objects should be considered dissimilar, then use the former transformation, or similar, then use the latter transformation. That is, the latter should be used when you want to consider highly negatively correlated objects as having a small distance. Non-informative genes contribute random terms in the calculation of distance. The resulting effect is that they hide the useful information provided by other genes. Therefore, non-informative genes should be assigned a 0 weight for use by the clustering algorithm. This translates to excluding those data points from the cluster analysis. Gene filtering is performed on expression data in order to remove, as much as possible, sources of obscuring variation. The filtering step may exclude spots or probe sets with too many bad values on the arrays, exclude spots or probe sets that do not vary significantly, or may exclude spots or probe sets with a fold change less than an arbitrary threshold. This is an important step prior to high-level data analysis because most algorithms used in such analyses do not perform well with thousands of variables. Following pre-processing steps, the resulting background-adjusted normalized data must be examined to identify which genes are most likely to yield insight into the biological processes being evaluated. There are general approaches for identifying such uninformative genes, such as computing some measure of variability, such as the coefficient of variation, which is the standard deviation divided by the mean, for each gene across all arrays, and then retain those that show the highest variability. Retaining genes whose absolute difference exceeds a predefined threshold, retaining genes whose expression is above a predefined minimum expression value. For Affymetrix gene chips, this may be redefined to retaining only those probe sets declared present using the detection call algorithm, or removing probe sets that are absent across all samples, or retaining only those well-characterized genes or probe sets using the most up-to-date annotation data. The authors of the seminal paper that applied clustering to microarray gene expression data developed a publicly available software program called Cluster, and corresponding a TreeView program. For some time, these were widely used, being among only a few available software tools for easily clustering microarray data. That software include the following filters. Keep genes or probe sets when the percentage of genes or probe sets declared present among all arrays exceeds a user-specified value. The default was 80%. Keep genes or probe sets for which the standard deviation exceeds a user-defined threshold. The default was 2. Keep genes or probe sets for which the maximum minus minimum expression values across all arrays exceeds a user-defined threshold. There are different clustering methods which fall into two broad categories. Hierarchical clustering, which includes agglomerative and divisive methods, and partitioning methods, which includes k-means and partitioning around medoids.